Um, Hello, this is Dr. Omar Amin Mohammed from the Department of Biology, Federal University of Health Sciences, Azari. And this is the second virtual lecture on General Biology 2, Bio 106. If you remember in our previous class, we were talking about the history of microbiology. And in this class, I'm going to categorize those important eras into four eras, which include the discovery era, the transition period, the golden age, and the development in medicine and surgery. Now, in a previous class, if you remember, we were talking about Zacharias Janssen, Robert Hooke, and Anton van Leeuwenhoek. They are classified into the discovery era. As you can remember, Zacharias Janssen is basically one of the first to be credited to making the earliest compound microscope, which was a couple of magnifying lenses. Then this paved way to development of a compound microscope that was done by Christopher Koch of the Coventry Garden, London, which was used by Robert Hooke to discover the cell. And after discovering the cell, another scientist came who is basically known as Anton van Leeuwenhoek, who was basically a Dutch line merchant, but he spent his time working on constructing simple microscope of double convex lenses, right? And he produced over 250 small powerful microscope that could magnify around 50 to 300 times the size of a small living organism. And then the next era is basically the discovery era, the transition era, sorry, which was the made up of the work of Francisco Redi, John Needham, and Lazzaro Spallanzani. And in this era is all about disproving and approving the theory of spontaneous generation, which is basically saying that living organisms arise from death and decay in myth. But this particular scientist, Francisco Redi, who showed that fly maggot do not arise from decay in meat, as others believe, if the meat is covered to prevent the entry of flies. Then John Needham, on the other hand, has another view. He advanced the spontaneous generation theory by trying to show that tiny microorganisms, their animal kills, arose spontaneously on the molten gravy or a meat or a broth. Then Lazzaro Spallanzani disputed the theory of spontaneous generation by boiling the broth, which is a piece of meat, in a soup or what have you, and showed that if you boil it, it will not arise or give rise to other microorganisms. Now, in the Golden Age, which is basically the beginning of microbiology as a subdiscipline of biology, the concept of spontaneous generation was finally put to rest by the French chemist Louis Pasteur. He inspired some set of experiment involving a gooseneck flask, and when he boiled the broth, which is a piece of meat, in a flask with a straight neck, and left it exposed to air, organisms grew on it. When he did this with a gooseneck flax, nothing grew. The gooseneck flax is a flax that is basically S in shape, right? And it shows that the flax trapped those particles from the air, preventing them from reaching the broth. By showing that he could allow air to get into the flax, but not the particle in the air, Pasteur proved that it was the organism in the dust that were growing in the broth. He also carried out, this actually put a rest and end to the debate of spontaneous generation from that particular time. He also carried out numerous experiments to discover why wine and dairy products became sour, and he found that bacteria were to blame. Now, Pasteur called the attention of the importance of microorganisms in everyday life and steered scientists to think that if bacteria could make the wine sick, then perhaps they could cause human illness and they could cause disease. This particular discovery contributed immensely to the development of the theory of the germ theory of disease, and this discovery and the discovery of Anton van Leeuwenhoek and the discovery of Koch and other scientists. And this particular theory states that microorganisms known as pathogens or germs can lead to diseases. So this made scientists to name Louis Pasteur as the father of modern microbiology and the father of bacteriology. That's the end of this class. Cheers. See you soon.